Hi everyone, this is Sinclair, a video podcast detailing the work and discoveries done by David Sinclair's lab group at Harvard Medical School. In this second episode, we'll be tackling questions like, why do we age? What exactly is aging? And is there a singular cause of aging? Now, at the surface level, aging is pretty obvious. We all know what it means to look old, sound old, and even smell old. But this is just half the story. Why exactly do we even age in the first place? And do we have to age at all? I'll hand it over to David to talk about the most basic question here. Why we even age in the first place. If you are taken aback by the notion that there is a singular cause of aging, you are not alone. If you haven't given any thought at all as to why we age, that's perfectly normal too. A lot of biologists haven't even given it much thought. Even gerontologists, doctors who specialize in aging, often don't ask why we age. Aging research today is at a similar stage as cancer research was in the 1960s. We have a robust understanding of what aging looks like and what it does to us, and an emerging agreement about what causes it and what keeps it at bay. From the looks of it, aging is not going to be that hard to treat, far easier than curing cancer. In the 1950s, the concept of group selection and evolution was going out of style, prompting three evolutionary biologists, J. B. S. Hallidane, Peter B. Medawar, and George C. Williams, to propose some important ideas about why we age. When it comes to longevity, they agreed individuals look out for themselves. Driven by their selfish genes, they press on and try and breed for as long and as fast as they can, so long as it doesn't kill them. In some cases, however, they press on too much, as my great-grandfather Miklos Vitas, a Hungarian screenwriter, proved to his bride 45 years his junior on their wedding night. If our genes don't ever want to die, why don't we live forever? The trio of biologists argued that we experience aging because the forces of natural selection required to build a robust body may be strong when we are 18, but decline rapidly once we hit 40, because by then we've likely replicated our selfish genes in sufficient measure to ensure their survival. Eventually, the forces of natural selection hit zero. The genes get to move on, we don't. Medawar, who had a penchant for verbiage, expounded upon a nuanced theory called antagonistic pleiotropy. Put simply, it says genes that help us reproduce when we are young don't just become less helpful as we age, they can actually come back to bite us when we are old. 20 years later, Thomas Kirkwood at Newcastle University framed the question of why we age in terms of an organism's available resources. Known as the disposable soma hypothesis, it is based on the fact that there are always limited resources available to a species: energy, nutrients, water. They therefore evolve to a point that lies somewhere between two very different lifestyles: breed fast and die young, or breed slowly and maintain your soma, your body. Kirkwood reasoned that the organisms can't breed fast and maintain a robust, healthy body. There simply isn't enough energy to do both. Stated another way, in the history of life, any line of creature with a mutation that caused it to live fast and attempt to die old soon ran out of resources and was thus deleted from the gene pool. These theories fit with the observations and are generally accepted. Individuals don't live forever because natural selection doesn't select for immortality in a world where an existing body plan works perfectly well to pass along a body's selfish genes. And because all species are resource limited, they have evolved to allocate the available energy either to reproduction or to longevity, but not both. That was as true for M. serperites as it was and still is for all species that have ever lived on this planet. All, that is, except one, Homo sapiens. Well put, David. Just recently, in evolutionary terms at least, we humans have managed to escape the literal rat race that all animals, including ourselves, once competed in for their very survival. Ironically enough, it is the origins of our species' success in our younger days that is now the downfall of it in our older days. Millions of years ago, cellular organisms had to choose survival in the now at the cost of survival in the later. What good would it do you if you could live to be 100 only to be eaten within the next hour? Simply put, our species did not evolve to live forever. We evolved to survive. And at a high level, this is the origin of aging. If you're unsatisfied with this answer, then you're just like me. In a later video, I'll be diving deeper into the origins of aging, narrowing down on the very genes that may be responsible for it. But for now, this big picture view will have to do. So now that we know a little bit about why we age from a big picture point of view, what about the little picture? Are there any theories that explain why we age at a cellular level? I'll hand it over to David again to give us some insight and share his knowledge on just those very theories. What is needed is a unified explanation for why we age, not just at the evolutionary level, but at the fundamental level. But explaining aging at a fundamental level is no easy task. It will have to satisfy all known laws of physics and all rules of chemistry, and be consistent with creatures of biological observations. It will need to span the least understood world between the size of a molecule and the size of a grain of sand. It should explain simultaneously the simple and most complex living machines that have ever existed. It should therefore come as no surprise that there has never been a unified theory of aging, at least not one that is held up, though not for lack of trying. The idea that mutation accumulation causes aging was embraced by scientists and the public alike in the 1950s and 60s, at a time when the effects of radiation on human DNA were on a lot of people's minds. But although we know with great certainty that radiation can cause all sorts of problems in our cells, it causes only a subset of the signs and symptoms we observe during aging, so it cannot serve as a unified theory. Around the same time that people were focusing on radiation, Dennin Harman, a chemist at Shell Oil, was also thinking atomically, albeit in a different way. After taking time off to finish medical school at Stanford University, he came up with the free radical theory of aging, which blames aging on unpaired electrons that whiz around within our cells, damaging DNA through oxidation, especially in the mitochondria, because that is where most free radicals are generated. Through the 1970s and 80s, Harman and hundreds of other researchers tested whether antioxidants would extend the lifespan of animals. The results were overall disappointing. Although Harman had some success in increasing the average lifespan of rodents, such as with the food additive butylated hydroxytoluene, none showed an increase in maximum lifespan. In other words, a cohort of study animals might live a few weeks longer, on average, but none of the animals was setting records for individual longevity. Free radicals do cause mutations. Of course they do. You can find mutations in abundance, particularly in cells that are exposed to the outside world, and in mitochondrial genomes of old individuals. Mitochondrial decline is certainly a hallmark of aging and can lead to organ dysfunction, but mutations alone, especially mutations in the nuclear genome, conflict with an ever-increasing amount of evidence to the contrary. Arlen Richardson and Holly Van Rimmen spent about a decade at the University of Texas at San Antonio testing if increasing free radical damage or mutations in mice led to aging. It didn't. In my lab and in others, it has proven surprisingly simple to restore the function of mitochondria in old mice, indicating that a large part of aging is not due to the mutations in mitochondrial DNA either, at least not until late in life. Although the discussion about the role of nuclear DNA mutations in aging continues, there is one fact that contradicts all these theories, one that is difficult to refute. If old cells had indeed lost crucial genetic information, and this was the cause of aging, we shouldn't be able to clone new animals from older individuals. Clones would be born old. It is a misconception that cloned animals age prematurely. It has been widely perpetuated in the media, and even the National Institutes of Health's website says so. Yes, it is true that Dolly, the first cloned sheep, created by Keith Campbell and Ian Wilmot at the Roslin Institute at the University of Edinburgh, lived only half a normal lifespan and died of a progressive lung disease. But extensive analysis of her remains showed no signs of premature aging. Meanwhile, the list of animal species that have been cloned and proven to live a normal, healthy lifespan now includes goats, sheep, mice, and cows. Because of the fact that nuclear transfer works in cloning, we can say with a high degree of confidence that aging isn't caused by mutations in nuclear DNA. Sure, it's possible that some cells in the body don't mature, and that those are the ones we end up making successful clones out of, but that seems highly unlikely. The simplest explanation is that old animals retain all the requisite genetic information to generate an entirely new, healthy animal, and that mutations are not the primary cause of aging. So what David is talking about here is pretty groundbreaking. He's basically laid out evidence refuting all previous theories of aging, some of which you've probably heard of. Namely, the theory that the accumulation of DNA mutations over time, caused either via external elements like free radicals or internal elements like replication errors, is the reason we age. As David said, this theory hasn't exactly panned out to be the reason why we age, and I think it's best refuted by the cloning example he gave at the end. For the DNA mutation theory to stand, it would be impossible to create a young clone from an old animal, as the starter cells harvested from the old animal would retain these mutations, resulting in an old clone. But as David said, this simply isn't the case. We can take cells from an old animal, like Dolly the sheep, and create a young clone from them. What this means is that at all points in your cell's life, they retain the genetic information of their younger self, of the younger you. Genetically, there is no difference between a 90-year-old cell and a 1-year-old cell. So, if this doesn't cause aging, then what does? Is there one singular cause of aging? I'll let David tell you for himself. Recently, the ideas of the leading scientists in the aging field began to coalesce around a new model, one that suggested that the reason so many brilliant people had struggled to identify a single cause of aging was that there wasn't one. In this more nuanced view, aging and the diseases that come with it are the result of multiple hallmarks of aging. Genomic instability caused by DNA damage, attrition of protective chromosomal end caps, the telomeres, alterations to the epigenome that controls which genes are turned on and off, loss of healthy protein maintenance known as proteostasis, deregulated nutrition sensing caused by metabolic changes, mitochondrial dysfunction, accumulation of senescent zombie-like cells that inflame nearby healthy cells, exhaustion of stem cells, altered intercellular communications, and the production of inflammatory molecules. Researchers began to cautiously agree. Address these hallmarks, and you can slow down aging. Slow down aging, and you can forestall disease. Forestall disease, and you can push back death. Each can be addressed one by one, a little at a time, in ways that can help us extend human health spans. There is little doubt that the list of hallmarks, though incomplete, comprises the beginnings of a rather strong tactical manual for living longer and healthier lives. Interventions aimed at slowing any one of these hallmarks may add a few years of wellness to our lives. If we can address all of them, the reward could be vastly increased average lifespans. As for pushing way past the maximum limit, addressing these hallmarks might not be enough. Although it is in its early days, a new shift in thinking is again underway. Still quite confident that the hallmarks are accurate indicators of aging and its myriad of symptoms, but unable to explain why the hallmarks occur in the first place. It is time for an answer to this very old question. Now, finding a universal explanation for anything, let alone something as complicated as aging, doesn't happen overnight. Any theory that seeks to explain aging must not just stand up to scientific scrutiny, but provide a rational explanation for every one of the pillars of aging. A universal hypothesis that seems to provide a reason for cellular senescence, but not stem cell exhaustion would, for example, explain neither. Yet I believe that such an answer exists, a cause of aging that exists upstream of all the hallmarks. Yes, a singular reason why we age. Aging, quite simply, is the loss of information. There are two types of information in biology, and they are encoded entirely differently. The first type of information, the type my esteemed predecessors understood, is digital. Digital information, as you likely know, is based on a finite set of possible values. In this instance, not in base 2 or binary, coded as zeros and ones, but the sort that is quaternary, or base 4, coded as adenine, thymine, cytosine, and guanine, the nucleotides A, T, C, and G of DNA. Because DNA is digital, it is a reliable way to store and copy information. Indeed, it can be copied again and again with tremendous accuracy, no different in principle from digital information stored in computer memory or on a DVD. The other type of information in the body is analog. Analog information is more commonly referred to as the epigenome, meaning traits that are heritable that aren't transmitted by genetic means. In the same way that genetic information is stored as DNA, epigenetic information is stored in a structure called chromatin. DNA in the cells isn't flailing around disorganized. It is wrapped around tiny balls of proteins called histones. These beads on a string self-assemble to form loops, as when you tidy your garden hose on your driveway by looping it into a pile. Epigenetic information is what orchestrates the assembly of a human newborn, made up of 26 billion cells from a single fertilized egg, and what allows the genetically identical cells in our bodies to assume thousands of different modalities. If the genome were a computer, the epigenome would be the software. It instructs the newly divided cells on what type of cells they should be and what they should remain, sometimes for decades, as in the case of individual brain neurons and certain immune cells. That's why a neuron doesn't behave one day like a skin cell, and dividing kidney cells don't give rise to 2 liver cells. Without epigenetic information, cells would quickly lose their identity, and new cells would lose their identity too. If they did, tissues and organs would eventually become less and less functional until they failed. But why do we have 2 types of information storage? Well, in the warm ponds of the primordial earth, a digital chemical system was the best way to store long-term genetic data. But information storage was also needed to record and respond to environmental conditions, and this was best stored in the analog format. Analog data are superior for this job because they can be changed back and forth with relative ease whenever the environment within or outside the cell demands it. And they can store an almost unlimited number of possible values, even in response to conditions that have never been encountered before. The unlimited number of possible values is why many audiophiles still prefer the rich sounds of analog storage systems. Even though analog devices have their advantages, they have a major disadvantage. In fact, it's the reason we've moved from analog to digital. Unlike digital, analog information degrades over time, falling victim to the conspiring forces of magnetic fields, gravity, cosmic rays, and oxygen. Worse still, information is lost as it's copied. Don't be disheartened by my claim that we are the biological equivalent of an old DVD player. This is actually good news. If it had been correct that DNA mutations were the main cause for aging, we would not be able to easily address it. Because when information is lost without a backup, it is lost for good. Ask anyone who's tried to play or restore content from a DVD that's had a corner broken off of it. What is gone is gone. But we can usually recover information from a scratched DVD, and if I am right, the same kind of process is what it will take to reverse aging. As cloning beautifully proves, our cells will retain their youthful digital information even when we are old. To become young again, we just need to find some polish to remove the scratches. This, I believe, is possible. And there it is, David's theory on why we age. A single reason, loss of information, brought about by various sources that results in aging. In the next episode, I'll be going over some everyday activities or behaviors that you can use to prevent or guard against the hallmarks of aging. You've certainly heard about these preventative measures like exercise or good nutrition as a way to stay healthy and fit, but their true potential to prevent aging has only recently been unveiled. If you'd like to stay up to date on David's latest works or support the channel, consider subscribing or following it on any of the social media platforms below. Thanks for listening, and until next time.